Very good afternoon to all the aspirants of EAPCET 2026 and NEET aspirants as well. We are in our last session of biomolecules, and here we are going to learn, as we have learned about nucleic acids in our previous session. Now we will exercise different forms of DNA and different forms of RNA. Of course, types of DNAs and RNAs are not there in your biomolecules chapter, but learning this here will help you with the chapter of molecular basis of inheritance. Right, Nama? Now you see we have three forms of DNA, that is aDNA, bDNA and zDNA. This is based on the helix sense, whether it is right handed or left handed, based on the major groove and minor groove distance, and based on the occurrence of the types of DNAs. Based on that, we have three different forms. First, if you see, first helix sense, okay, right handed DNA. This is right handed DNA, A helix and B helix. Both are A type and B type DNA, both are right handed DNA, whereas Z type DNA is left handed. Remember this, very important. And next, it is most important to know base pairs per turn. One turn or helix, one complete turn of DNA. This is one complete turn of DNA. This is addressed as one pitch or one turn. One pitch or one turn. One pitch or one turn of DNA will have 33.4 angstroms or 34 nanometers length of DNA. Okay, and in B type DNA, approximately it will have 10 base pairs. In one complete turn of DNA, you will have 10 base pairs of DNA in B form. Okay, and here you see A type DNA, hardly you will have 11 base pairs. B type you have 10.5 or approximately 10 base pairs of DNA. Z type will have 12 base pairs of DNA. Important note, remember. Next, major groove and minor groove. Major groove in B type DNA will be wide and deep, and here major groove will be flat or completely absent in zDNA. Major groove is narrow and deep, you can see here, right, narrow and deep in A type of DNA. Minor groove, you observe, it is wide and shallow in A type DNA, narrow and deep, can you see here, narrow and deep in B type DNA, same the ways, narrow and deep in Z type DNA as well. Now, if you observe the occurrence, B type DNA will occur mostly in normal physiological DNA is what? B type DNA only, and A type DNA is observed in dehydrated DNA or in the low waters. In high salt concentrations, special sequences will be present. These are observed in Z type DNA. And then biological relevance. Biological relevance, less common, most common will be B type DNA in the cells, and Z type DNA, they will play a role in gene regulation, and most stable DNA amongst these three types will be B type DNA, and less stable will be A type, least stable is Z type, because here DNA is arranged in zigzag fashion. Fine, right? It is left handed and it is arranged in zigzag fashion. Till here it is clear right now, and then you observe highly B type DNA characteristic features are more important, because the biological DNA is what? B type DNA. Just remember the comparative analysis between A, B and Z, because there is huge expectation, like we may expect match the following type of questions from types of DNA, not just in EAPCET but also in NEET. If you want, you can take a screenshot of this as well. Fine, right? Yeah. Now we will move to the next segment, that is Chargaff's rule. Chargaff's rule, which are also considered as the silent features of the DNA. Chargaff's rule is from what? Double helical model of DNA. Yes or no, ma? From double helical model of DNA we got this Chargaff's rule. So in 1950s, Fine, right. In 1950s, it is the scientist Erwin Chargaff. He has concluded that DNA molecule, in any DNA molecule, the number of pyrimidines include cytosine and thymine, will be equal to number of purines. Number of pyrimidines will be equal to number of purines in any DNA molecule. That is, A plus G is equal to C plus T. Fine, right? A plus G is equal to T plus C. The amount of adenine will always be equal to amount of thymine, and amount of guanine will always be equal to amount of cytosine. And from silent features we have learned, right, how many hydrogen bonds will be present between adenine and thymine? It will be 2 hydrogen bonds. How many hydrogen bonds between cytosine and guanine? It is 3 hydrogen bonds. Fine, right? And now you see base ratio. Base ratio is nothing but A plus T by G plus C. Fine, right? A plus T by G plus C will give you a base ratio. This base ratio will vary from one species to other species. In prokaryotic cells, like E. coli, for example, E. coli, base ratio will be approximately 0.9, that is less than 1. Base ratio will be less than 1 in prokaryotic cells. In eukaryotic cells, if you assume, base ratio will be more than 1. Base ratio will be more than 1 in eukaryotic cells. It will be less than 1 in prokaryotic cells. So, here the deoxyribose sugar and phosphate group, components occur in the equal proportion, like amount of sugars, like number of sugars will be equal to number of phosphate groups that are present. Remember that. Fine, right? So, this will give you the complete set of what? Chargaff's rule. Fine, right? Now, yeah, here we go with types of RNAs. We have 3 different types of RNAs like mRNA, rRNA and tRNA. What will be the linkage of RNA here? RNA is linked by the same bonds, right? One nucleotide is connected to other nucleotide by phosphodiester bond only. See, this is how 2 nucleotides are connected. Can you see here? Yeah, those are connected by what? Phosphodiester bond. That is 3' to 5' connected. Phosphodiester bond. Fine, right? And you have different nitrogen bases will be adenine, uracil, guanine and cytosine. In a single nucleotide, again you will have same bonds like N-glycosidic bond and phosphodiester bonds. Now, we will look into the synthesis. Where are these synthesized in a eukaryotic cell? Let us suppose this is the eukaryotic nucleus. Eukaryotic nucleus, let us suppose. Just a cartoon diagram. And inside which you have nucleolus. Fine, right? So, here messenger RNA and tRNA are synthesized inside the nucleoplasm. Fine, right? Messenger RNA and tRNA are synthesized in nucleoplasm. Like tRNA synthesized by RNA polymerase 3. mRNA is synthesized by RNA polymerase 2. Fine, right? mRNA synthesized by RNA polymerase 2, tRNA synthesized by RNA polymerase 3 inside the nucleoplasm. Whereas inside the nucleolus, RNA polymerase 1 will be synthesizing rRNA, that is ribosomal RNA. Important. Of course, these are not present in biomolecules, but this knowledge will help you in easy understanding of molecular basis of inheritance. So, have a note of this. Now, we will look on to messenger RNA. You see, messenger RNA, what is the function that it is doing? This carries genetic code from DNA to ribosome, and this is present just 5%. 5% of total RNA is your messenger RNA, followed by ribosomal RNA. Ribosomal RNA, whose function is what? Structural and enzymatic role in ribosomes. rRNA is present, like 80% of total RNA is rRNA, followed by tRNA, which is also known as soluble RNA and adaptive RNA. Okay, tRNA also addressed as soluble RNA and adaptive RNA. This helps in transfer of amino acids to ribosomes during translation. How much percent it is present, ma? 10 to 15% of total RNA is what? tRNA. 10 to 15% of total RNA will be tRNA. Clear till here? Types of RNA, its functions and its total proportion in the cellular RNA. Till here it is fine, right, ma? Yeah. Next we will move on to lipids. Nucleic acids is done, proteins is done, and carbohydrates are done. Now the last macromolecule, that is lipid, which is not strictly a biomacromolecule, because its molecular weight will be less than, its molecular weight will be less than 800 Daltons. Fine, right? They are generally what? Water insoluble. Lipids are usually water insoluble. Yeah. They are generally water insoluble. They could be simple fatty acids, or a fatty acid that has carboxyl group. The general formula is like RCOH. This R group can be any functional group. It can have n number of carbon groups, like it is general formula is RCOH. It has one carboxyl group attached to R group, the general formula of lipids. Okay. Next, the R group could be methyl group or ethyl group, or highest number of carbons can also be present, like 1 carbon to 19 carbons. For example, if you see, palmitic acid will have 16 carbons including carboxyl carbon. Including carboxyl carbon, it is having what? 16 carbons for palmitic acid. Arachidonic acid, if you observe, it has 20 carbons including carboxyl carbon. Including carboxyl carbon, how much it is having, ma? 20 carbons. Till lipids it is fine, right? Next you look into simple lipids. Simple lipids, we have these types, like neutral or true fats. Yeah. Simple lipids, conjugated lipids, derived lipids. We have 3 types of lipids. Let us exercise it one by one. First is neutral or true fats under simple lipids. These are esters of fatty acids. What are these neutral fats, ma? They are esters of fatty acids with glycerol. So they are addressed as what? Glycerides. They are addressed as glycerides. Here we learn it with an example, glycerol. Glycerol will have how many carbons? Okay, it is having 3 carbons, each bearing one hydroxyl group. So it is known as what? Triglyceride or trihydroxypropane. Remember the IUPAC name. It is known as what? Glycerol, triglyceride or trihydroxypropane. Clear with neutral or true fats? Fine, right? It has a carboxyl group and a carbon group. Fine, right, ma? Okay, yeah. Hydroxyl group is present, which is attached to carbon group. Next, fatty acids could be saturated fatty acids or unsaturated fatty acids based on the bonds, number of bonds which are present in between carbons. Okay, which are present in between carbons, we have saturated and unsaturated. Saturated fatty acids will have no double bond between them, they are without a double bond, whereas unsaturated fatty acids are with one or more carbon-carbon double bonds. Okay, they are with one or more carbon-carbon double bonds. Here you see saturated fatty acids. Fine, right? A fatty acid molecule, any fatty acid molecule is unbranched chain of carbon atoms. It is unbranched chain of carbon atoms. This has carboxyl group attached to R group, like what we have learned. Carboxyl group attached to R group. So along with R group, along with carboxyl group, it can be 16 carbons like in palmitic acid, 20 carbons like in arachidonic acid. This will be the representation for what? Palmitic acid and stearic acid as well. Stearic acid along with carboxyl group, it has 18 carbons. Fine, right? Now we'll move on to unsaturated fatty acids. What are the examples of unsaturated fatty acids? Remember, it is very important to know about the examples of saturated fatty acids and unsaturated fatty acids. Here you look into unsaturated fatty acids. Fatty acids with one or more double bonds. Here it has one or more double bonds. Examples you see oleic acid, linoleic acid, linolenic acid and arachidonic acid. These are the examples of what? Unsaturated fatty acids. Many lipids will have both glycerol and fatty acids. They will have both glycerol and fatty acids. Here fatty acids are found esterified with glycerol. They can be monoglycerides or diglycerides or triglycerides. Fine, right, ma? Example you see here oleic acid and linolenic acid. Here you have double bonds in between them, in between carbons. So these are what? Unsaturated fatty acids. Now you see about glycerides. Many lipids with glycerol are fatty acids. They are fatty acids. Here the fatty acids are found esterified glycerol, like you see. Yeah, glycerol and 3 fatty acids. It is triglyceride. One glycerol with 2 fatty acids. It will be diglyceride. One glycerol with 1 fatty acid. It is monoglyceride as such. Okay, fine. Clear till here? Yeah. Now, based on the melting point of glycerides, triglycerides can be called as fats or oils, like this. They can be considered as fats or oils. Fats will have high melting point, they will be having high melting point and they remain as solids at room temperature. They will remain as solids at room temperature, for example, butter and ghee. Oils will have low melting point and they remain as what? Liquids or oils at room temperature, for example, gingerly oil and sunflower oil. This is about normal simple lipids. Now we will look on to derived lipids. Okay, derived lipids, for example, steroids. The steroids do not contain fatty acids. Remember, steroids doesn't contain any fatty acids, but they are included in lipids because they have fat-like properties. Okay, these derived lipids are having fat-like properties. They have most common steroids, for example, sterols. A common sterol can be cholesterol. Okay, so cholesterol is the most abundant steroid in animal tissues. Food rich in animal fats contain what? Cholesterol. It is also synthesized in liver. Fine, right? Cholesterol is essential component of animal plasma membrane, which will maintain stability and fluidity of the plasma membrane. Fine, right? This will maintain stability and fluidity of plasma membrane. Till here everything is clear, right ma? You can take a screenshot highlighting the steroid properties. Now, we will move on to the next concept, that is compound or conjugated lipids. Important ma. Yeah. What are compound or conjugated lipids? Usually fatty acids or simple lipids, they are having a functional group and carboxyl group. Along with functional group and carboxyl group, if they are consisting of any other components like phosphate groups or alcohol groups or sugar components, they are addressed as what? Compound or conjugated lipids. Here you observe, first type is what? Phospholipids. First type is phospholipids. Phospholipids components include glycerol or any other alcohol, like it has two fatty acids. Phosphate group will have a colon nitrogen containing group. For example, lecithin. Important. Quite a times we got question on lecithin. So, here what are the components it has? Glycerol or other alcohol, two fatty acids along with phosphate group and colon. Fine, right? Where is this type of conjugated lipid present? These they form cell membranes, amphipathic molecule which has both polar hydrophilic head and non-polar hydrophobic tails. Polar hydrophilic head and non-polar hydrophobic tails. Now, glycolipids. Glycolipids contain fatty acids like alcohol and sugar. Okay, they will have fatty acids, alcohol and sugar. Okay, only fatty acid alcohol you will address it as sphingosine. Along with sugar, you will call it as, for example, you can take it as galactose. Now, you see functions of this. Sugar replaces one fatty acid which is present in myelin sheath of nerve fibers and chloroplast outer membrane as well. And then if you see lipoproteins. Jagadishwari, just ask it in a proper way ma. Can it be broken like fatty acid by changing any physically? The doubt is not clear, please ask it in a proper way, then it will be clarified, right? Yeah. Now, lipoproteins include what? Lipids, mainly phospholipids, and it has proteins, fine, right? Lipids along with proteins. What conjugated lipid it is? Lipoprotein. They are usually making up the structural component of the plasma membranes. This will transport lipids into the blood. Now, chromolipids. Fine, right? Chromolipids include pigments like carbohydrates. Okay, they will include pigments like carbohydrates. Here, pigmented lipids are involved in coloration and photosynthetic process. Examples you can take is keratin and vitamin C. These are the examples of what? Chromolipids. Fine, right? You can take a screenshot of this. Yeah. Let's continue with the session. So, these are the structures which are given in your NCERT. See, 16 carbon including carboxyl group, palmitic acid, which is the example for saturated fatty acids. Triglyceride or glycerol, fine, right? 3 hydroxyl groups are present over here, and this is triglyceride. You can see 3 carboxyl groups also, fine, right? And phospholipids like lecithin is present over here, phospholipid, which is the major component of cell membrane. Cholesterol, this is the example of what? Steroids or derived lipids. Derived lipids or steroids. Till here it is clear, ma? Now, metabolism will directly go to enzymes, right? Yeah. Enzymes, most of the enzymes are what? Proteins, but few of the nucleic acids will also behave like enzymes. Example is ribozyme. Okay, ribozyme is the best example where nucleic acid is behaving like enzyme. Enzyme can be addressed as many ways. Best way of addressing enzyme is the biocatalyst. Enzymes are biocatalysts, which will enhance the rate of biological reactions that are happening inside the living organism. Here if you see, enzymes are organic catalysts. They are inorganic catalysts also. There are some inorganic catalysts also, but they do not occur inside the living cells. Fine, right? So, here there are clear differences between enzymes and inorganic catalysts. What are the clear differences between enzymes and inorganic catalysts? Just go through. Fine, right? If you see here keenly, enzymes are what? Complex biological macromolecules. Fine, right? Complex biological macromolecules which are made up of proteins. Inorganic catalysts, they are usually simple or small sized molecules or elements. Here these catalysts will operate within the living organisms only, that is within the living systems. Here these are found and they function in non-living systems, that is outside the living cell. Each enzyme is highly specific for every single biochemical reaction. Fine, right? Here a single inorganic catalyst can act for multiple different reactions. Here there is no specificity for inorganic catalysts. Enzymes lose activity at temperature which is above 40 degrees Celsius, but here they remain stable and work efficiently even at high temperatures and pressures because the enzymes are thermophilic in inorganic catalysts. And here enzymes catalyze reactions with remarkable speed and precision. Here inorganic catalysts generally work slower with lower specificity, mostly they are non-specific. These are the key differences between enzymes and inorganic catalysts. Now we will move on to the enzymes which are from thermophilic organisms. These are thermally stable. Like if you see, these are biocatalysts only, whichever are derived from thermophilic organisms. They will work efficiently at high temperatures like up to 80 to 90 degree Celsius. Now we will look on to mechanism of enzyme action. First you see briefly how enzymes will act. What is the mechanism of enzyme action? Any enzyme, let us suppose sucrose as the substrate, which is given as such in this diagram. Yeah. Let us suppose sucrose as the substrate. Fine, right? This sucrose substrate, by the activity of the enzyme, that is sucrase. Okay, sucrase is the enzyme which is acting on this. This will give out two products. One is glucose and one is fructose. So here what is the substrate, ma? Sucrose is the substrate. Over the substrate, some active sites will be there. To these active sites, over the enzyme, active sites will be there. To these active sites, the substrate will come and bind. Fine, right? Just a second. Fine, right? Enzyme is having what? Active sites. To this active sites, the substrate will come and bind. Fine, right? Sucrose is the substrate which is coming and binding to the active site of the enzyme, sucrase. Now, this forms an enzyme substrate complex. Okay? Enzyme substrate complex, where enzymes will work on the substrate vigorously and convert it into enzyme product complex. Enzyme substrate complex, to be converted to enzyme product complex, requires highest potential energy. Highest potential energy, which is addressed as transition energy. This is addressed as transition energy. Fine, right? So, here first enzyme which is having active sites, to this active site substrate will come and fit. How the substrate is fitting? We have two models, lock and key hypothesis and induced fit model. We will learn it, and as such it is forming what? Enzyme substrate complex. Enzyme substrate complex will be modified to enzyme product complex. Here, to convert or to transit enzyme substrate to enzyme product, this requires highest potential energy, which is also addressed as transition energy. Okay, this is transition energy from enzyme substrate to enzyme product, and this will give out two products. Fine, right? Glucose and fructose. As such, the products will be released from enzyme product complex. The products will be released. Fine, right? So, the transition energy to convert from enzyme substrate to enzyme product is highly minimized. The energy is highly minimized with the enzyme. That is addressed as activation energy, which is the minimal energy required to transit from enzyme substrate to enzyme product. Fine, right? Usually it is transition energy, but in the presence of enzyme, minimal energy is required, that is known as activation energy. This is a brief idea that you have to know before getting into the models of lock and key hypothesis and induced fit model. Just go through this concept, just analyze this briefly. If you are having any doubts, drop a message in the chat box. I will address your doubt. Fine, right? Now, here we move on to lock and key hypothesis, lock and key model. See, lock and key model, yeah. Fine, right? Lock and key model is not that applicable for the biological systems, because the active site on the enzyme is addressed as lock site, where key is binding, which is very rigid. So, the key is fit into the lock, as like enzyme is addressed as lock over here and the key is substrate. This model is not applicable to the biological system. Lock and key hypothesis is given by M.L. Fisher. Next, you see induced fit model. In induced fit model, this is also known as induced fit model, wherein the active site of the substrate is not rigid like a lock here. The structure is adjusted according to the substrate, like how a hand is fit into the glove, same ways the substrate is fit into the active site of the enzyme, post which it forms enzyme substrate complex and then turns to enzyme product complex. This is more realistic model when compared to lock and key hypothesis. Now, we will look into the clear-cut differences between lock and key hypothesis and induced fit model. Yeah. Here we go. First, lock and key hypothesis is proposed by whom? M.L. Fisher, right? It is proposed by M.L. Fisher. Induced fit model is proposed by Daniel Koshland. The concept says like enzyme active site is exactly complementary to substrate's shape, but here active site will change the shape to fit the substrate by binding. Analogy, if you see, the key is substrate which fits into the lock, that is enzyme. Here hand is addressed as substrate which fits into the globe, that is enzyme. Flexibility of the enzyme, enzyme structure is rigid and it does not change. Here enzyme structure is flexible and this adjusts to the substrate. Fine, right? Next, lock and key hypothesis. This is highly specific, high specificity due to perfect fit of lock and key, but this is from moderate to high specificity where the fit is optimized according to the interaction between enzyme and substrate. Next, adaptability to the substrate. This cannot explain the enzyme binding to similar shaped structures, but induced fit model will explain the binding to a range of similar substrates. Clear, right? Next, scientific acceptance. Lock and key hypothesis is older model. This is considered limited. Induced fit model is more accepted and realistic model. Just take a screenshot of this comparative analysis, it is very important. And next, we will move on to the properties of enzymes. What are the properties of enzymes? Those are biocatalysts. Also addressed as what ma? Organic catalysts. Here we go. First property is protein nature. Enzymes are generally proteinaceous. They may require inorganic or organic substances for the activity. Next, chemical reactions. Enzymes do not initiate reactions, but they increase their rate. They are not alter chemical equilibrium. Next, efficiency. Efficiency is directly equal to turnover number, that is turnover number, where substrate molecules are converted per minute per enzyme molecule. Next, enzymes will remain in unchanged form after the reaction as well. Enzyme specificity. Enzymes are highly specific to their substrates. For example, maltase is specific for the substrate maltose, not sucrose. For sucrose, what is the specific enzyme? Sucrase. As such, these are the major properties of the enzymes. Now, enzyme catalysts will differ from inorganic catalysts that we have learned. And now chemical reactions. How you can identify rate of chemical reaction? Rate of chemical reaction can be defined by amount of product by time taken. Okay? Amount of products formed by unit time or amount of products formed per unit time. This will give out what? Rate of any chemical reaction. And there will be huge influence of the factors like temperature and pH for the chemical reaction. Like if you see, chemical reaction is influenced by temperature and rate will double or decrease by half. They will double or decrease by half for every 10 degrees Celsius rise or fall of the reaction. For every 10 degrees Celsius rise or fall of the reaction, the rate of the reaction will be raised or dropped for every 10 degrees Celsius rise. Fine, right? So, these are the activities of rate of reaction. How you can identify rate of reaction? Amount of product formed per unit time. What is the influence of temperature? For every 10 degrees rise, the activity chemical reaction rate will be doubled. For every 10 degrees Celsius drop, the reaction rate is put to half. Now, carbon dioxide and water. In the presence of the enzyme, carbonic anhydrase, this forms carbonic acid. Okay? Carbon dioxide along with water in the presence of carbonic anhydrase, this forms carbonic acid. In absence of any enzyme, this reaction is very slow. Okay, this reaction is very slow in the absence of any enzyme, with about 200 molecules of hydrogen carbonate being formed in single hour. By using the enzymes carbonic anhydrase, the reaction speeds up dramatically with about 6 lakh molecules formed per second. Okay, 6 lakh molecules formed per second. The enzyme has accelerated the reaction rate by about 10 million times. Fine, right? Remember this, it is very important. These are the direct liners of your NCERT. Here we go with the graph, most important one. So here I will give you two types of reactions. Based on the graph you can analyze whether it is exothermic chemical reaction or endothermic reaction. Or endothermic reaction. Here if we see maximum potential energy required to convert from substrate to product, that is the conversion from enzyme substrate to enzyme product. Maximum potential energy required is addressed as transition energy, which is maximum potential energy, right? So in the presence of enzymes, this maximum potential energy is not required. With minimal energy only, enzyme substrate complex is turned to enzyme product complex. That energy is addressed as activation energy. Okay, it is activation energy which will convert from enzyme substrate to enzyme product complex. So here if substrate potential energy is high and product potential energy is low, which means that the energy is lost in the form of heat. The energy is lost in the form of heat when substrate is converted to product. Fine, right? When substrate is converted to product, energy is lost in the form of heat. This is addressed as what? Exothermic reaction. But here substrate potential energy is high and product potential energy is low. In endothermic reaction, substrate potential energy will be low and product potential energy will be high. Product potential energy will be high and substrate potential energy will be low, where heat or energy is consumed. Energy is consumed here. So from substrate to product, it is having high potential energy and this will have low potential energy. Clear? I hope you got a clear idea on transition energy, activation energy, exothermic reactions and endothermic reactions as well. Clear till here, ma? Any doubt anywhere? Just go through this. Fine, right? Next, factors affecting enzyme activity. For any biocatalyst, the factors which affects the enzyme activities include temperature, pH, and most important, substrate concentration. They play crucial role in the enzyme activity. If you see temperature, as we have discussed previously, for every 10 degrees rise in temperature, the enzyme activity is doubled. For every 10 degrees Celsius fall in temperature, the enzyme activity gets to half. That is the role of temperature, that is how it will affect the enzyme activity. And here if you see pH, fine, right, pH. Each enzyme will have a significant or specific pH. pH is not common for all the biocatalysts. It is specific for each enzyme. If you can see here, these are different enzymes that you will come across the NCERTs. For example, pepsin. This digests what? Proteins, whose optimum pH is 2. Fine, right? Trypsin, whose optimum pH is 8. Salivary amylase 6.8 to 7, which is present in saliva. Lipase, whose enzyme activity specific or optimum pH is 7.5 to 8. Maltase, optimum pH is 6 to 7. Rennin or chymotrypsin, whose optimum pH is up to 3.5 to 4. DNA polymerase, optimum pH is up to 7.5 to 8. Catalase, optimum pH is up to 7.0 is the optimum pH. So, pH is specific for each biocatalyst based on their location and enzymatic activity. Clear, right? If you want, take a screenshot of this, this will be helpful to you. Fine, right, ma? Now, factors affecting enzymatic activity. These include temperature, pH, change in substrate concentration or binding of specific chemicals. If you see temperature and pH I have already discussed. Now, substrate concentration. Both temperature and pH will give you a bell-shaped graph that shows drastic increase in enzymatic activity until it reaches optimal temperature and pH, and drastic downfall once after reaching the optimal temperature and pH range. Then you will see drastic downfall with increase in temperature or increase in pH. If you see for concentration, substrate concentration, this shows a curve like this, wherein as you increase the substrate concentration, velocity of the reaction will increase, velocity of the chemical reaction will increase. But with maximum increase, this gets to a saturation point. Okay, the reaction gets to a saturation point. For example, if I tell you there are 10 enzyme molecules, and we are adding on the substrates to it, like 1, 2, 3, as so on, we have increased up to 10. Till 10 it will increase the rate of reaction, after which even if you provide the substrate molecules, even if you provide the substrate, the rate of reaction comes to a saturation because enzymes are limited. As such, the velocity maximum is the range where maximum substrate concentration is bared by the enzyme. Half of the Vmax is addressed as substrate concentration. In Michaelis-Menten constant, Km is known as Michaelis-Menten's constant. There is a significant or specific formula, I can say, most of the times used, where Michaelis-Menten constant Km will be equal to half Vmax, half of the velocity which is attained by maximum substrate concentration. So, Km is always equal to what, ma? Half Vmax, that is Vmax by 2. Remember this formula. This is about substrate concentration. Now, about inhibition of enzymes. We are left with inhibition of enzymes and cofactors. Just hold on for 15 minutes, this will be clear to you. We have 3 patterns of inhibitory activities of the enzymes. One is competitive inhibition, second is non-competitive, third is allosteric or feedback inhibition. First, I will explain you 3 different modes, like what are the types of inhibitions. Competitive inhibition is, here inhibitor will compete with substrate to bind to the active site of the enzyme. Fine, right? Here the competition is in between inhibitor and substrate to bind to the active site of the enzyme. Non-competitive inhibition, here inhibitor will come and bind to the enzyme. It will change the active site shape. It will change the shape of active site of the enzyme, which will make the enzyme incapable or inactive to bind to the substrate. It will make the enzyme incapable or inactive to bind to the substrate. Fine, right? Allosteric or feedback inhibition. If products are produced at the maximum concentration, these products will inhibit the rate of the reaction of the first step. Fine, right? If the products are produced or the products are present at the maximum concentration, that will inhibit the rate of the reaction of the primary step. That is allosteric or feedback inhibition. Till here it is clear, right, ma? Now we see competitive inhibition. This binds to the active site. Here binding of the inhibitor is other than the active site. This here inhibitor to a regulatory or allosteric site. Structural similarity. Inhibitor is structurally similar to the substrate in competitive inhibition. Inhibitor cannot be structurally similar to the substrate. Here product or inhibitor, this may or may not resemble to the substrate. Fine, right? Here effect on Vmax. Vmax velocity, maximum velocity remains unchanged. Here Vmax is decreased because of the inhibitory activity. Here allosteric inhibition, the inhibitor will increase or decrease based on the modulator. Here product itself acts as inhibitor. Next, effect on substrate concentration. Substrate concentration is increased here. Here it is unchanged. Here it is variable in allosteric inhibition. It is very important to know the changes of maximum velocity and substrate concentration. Clear, right? And then, yeah, here we move on with examples. Here examples are important, ma. Yeah. For example, malonate will inhibit succinate dehydrogenase in what? Tricarboxylic acid cycle. Here non-competitive inhibition, best example cyanide, which inhibits cytochrome oxidase, which is complex 3 in electron transport chain. Next, here ATP will inhibit phosphofructokinase activity. This happens in glycolysis. Isoleucine will inhibit threonine deaminase. Fine, right? These are the examples that you have to remember. Now we will move on to enzyme classification. Enzymes are classified into 6 classes. Here for each class you should remember the examples which are very important and the function. First class of enzyme, if you see, first class of enzyme, oxidoreductases and dehydrogenases. Examples you see, dehydrogenase, oxidase, reductase, catalase and peroxidase. Functions, this will catalyze oxidation reduction reactions involving transfer of electrons or hydrogen from one molecule to other. Fine, right? These are oxidoreductases or dehydrogenases and these are the examples. Next is transferases. This will catalyze the transfer of specific functional groups like phosphate or amino group from one molecule to other. Examples you take, ma, transaminase, this will transfer amino group, kinase will transfer what? Phosphate group. Third is hydrolases. This will catalyze the breakdown or hydrolysis of larger molecules into smaller ones using water. Examples, protease, amylase, lipase, maltase and nucleases are the examples. Next, lyases. This will catalyze cleavage of substrate into 2 parts without using water. This forms double bond at cleavage site. Double bond is formed at cleavage site. Examples, aldolase, decarboxylase and carbonic anhydrase. Next is isomerases. We have isomerases, for example. This will catalyze rearrangement of molecular structure to form isomers. Example is isomerase. Isomerase is the example. Next is ligases. This will catalyze the joining of two molecules using ATP as energy source, using ATP. This forms covalent bonds like carbon oxygen, carbon sulfur, carbon nitrogen and phosphate oxygen. Examples you see RUBP carboxylase, ligase, phosphoenol pyruvate carboxylase. These are the examples. This is the complete set of classification with functions and examples. Very important, ma. Take a screenshot of this. Next is cofactors. What are cofactors? Okay, usually a functional enzyme will have both protein part and non-protein part. So here you can see protein part and non-protein part of the enzyme. Okay, non-protein part of the enzyme is addressed as prosthetic group. Protein part of the enzyme is known as apoenzyme. This prosthetic group can be cofactor, it can be metallic ions or coenzymes. First you see cofactor. These are non-protein constituents called as cofactors bound to the enzyme, which will make them catalytically active. Three kinds of cofactors: prosthetic group, coenzymes and metal ions. The protein portion of the enzyme is addressed as what? Apoenzyme. Here peroxidase and catalase, they will catalyze the breakdown of hydrogen peroxide is broke to water and oxygen. Heme group is a prosthetic group and it is a part of active site of the enzyme. I get fine, right? Next is coenzyme. Coenzymes are also organic compounds, but their association with apoenzyme is only transient. Apoenzyme is only transient. This usually occurs during course of analysis. The essential chemical components over here, essential chemical components of many coenzymes are what? Vitamins. And now coenzymes will serve as cofactors in a number of different enzyme catalyzed reactions. Coenzyme, if you see, coenzyme nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate. Okay, this contains vitamin what? Niacin vitamin is present. Lastly, metal ions. Metal ions are present, are aligned along with enzymes by coordination bonds. Okay, by strong coordination bonds. Here you see the number of enzymes will require metal ions of their activity to form coordination bonds with the side chains of the enzymes at the active site, and at the same time this form one or more coordination bonds with the substrate. For example, you see zinc is the cofactor for the proteolytic enzyme, that is carboxypeptidase. Okay, carboxypeptidase, zinc will be the cofactor. Clear till here? Any doubt anywhere, ma? Yeah. So with this, complete biomolecules chapter is done. If you are having any doubt, you can drop your message in the chat box. I will be addressing all your doubts. With this, we are done with cell biology, and next we will move on to reproduction in plants. Fine, right? So that's all for today's session, ma. Thank you.